Okay, good afternoon, everyone. Let's move to the next section. Today, I'll be talking about, I hope in the last part, I briefly talked about the patterning in the invertebrates, the Drosophila. Today, I'll be talking about the vertebrate. Mostly, today's lecture talking about vertebral patterning, anterior-posterior patterning, and dorso-ventral patterning. So we'll cover this one first, then I will move to the next dorso-ventral patterning in the second session. So before going detail about anterior-posterior patterning in vertebrates, I just want to kind of orient you how the brain develops and everything I have written in the notes so that it will be easy for you to follow whatever I'm saying. So the vertebrate brain develops from the neural tube. So this is one stage of the development. This is another stage, and this is the mature. This is another stage. So this is the latter stage of the embryonic development in mouse. So so this brains develop as a swelling from the neural tube, and you have three kind of vesicle kind of structure present in this in the earlier stage of the development. You have the prosencephalon, which is denoted in the kind of red or pink, I think red color. And you have the green color, the mesencephalon, the midbrain, and you have the metencephalon. So they develop as three different vesicles kind of structure. And latter stage of the development, the prosencephalon again divides into two distinct compartments. One is the telencephalon, anterior part, and you have the diencephalon that develops from the prosencephalon. The mesencephalon remain like same, and you have the another the hindbrain, which is called as rhombencephalon. So the rhombencephalon divides into metencephalon and myelencephalon. So these are the different segments or different parts of the brain which I will be covering about. And mostly the segmentation that I talked about in the last class about Drosophila, like Drosophila body, you have the segmentation, and there are specific set of gene that actually identifies or represents different segment. You have the segment polarity gene, you have the homeodomain gene, the homeobox gene in Drosophila. So similarly, more or less in vertebrates also, you have similar kind of segmentation found in the vertebrates, mostly in the hindbrain, which is the rhombencephalon. So the segmentation takes place in the hindbrain. Also, I'll be talking about segmentation kind of thing in the forebrain, the latter part of the lecture. So each segments called as rhombomeres, or each units in the rhombencephalon is called as rhombomeres. And as I mentioned earlier, the rhombencephalon is divided into two parts. One is the metencephalon, another is the myelencephalon. Okay, and set of genes required for rhombomere development. So the rhombencephalon is segmented, and you have rhombomeres. So, and each set of gene is required for the rhombomere development. I'll be talking about soon. Going back to the previous slides, so Hox genes. So in the Drosophila, you have Hox genes that defines the segmentation in the Drosophila, and this Hox gene basically defines the regional identity of the animals. And one group of Hox gene present in Drosophila, and these are represented here, and these are already color coded. So you have the antennopedia complex here, and you have the ultra-vithorax complex here. The antennopedia complex develops the anterior part, like here the labium part is also color coded here. And your different sets of genes are present in this cluster, which develops the anterior part of the animal. And you have the posterior part, the abdomen, and some part of the leg also develops from the ultra-vithorax complex. So like similar like in Drosophila, in vertebrates, you have four sets of genes, and you don't have to remember these genes. So don't have to remember what genes is required for like the segmentation. You don't have to remember. I have written in the slide. So you have 4 sets of gene like clusters, Hox genes cluster, like Hox A, Hox B, Hox C, and Hox T. This slide is just to revise you kind of this type of genes also present in the vertebrate, like I have discussed before, like in vertebrates. So we'll mostly talk about this is the example of a chick embryo, 3-day chick embryo. So mostly I'll be talking about the hindbrain segmentation, which I have talked in the previous slide, the rhombomeres development from the hindbrain. So here the chick embryo looks like this. You have the forebrain, the prosencephalon, and the midbrain, the mesencephalon, and the hindbrain, the rhombencephalon. Again, rhombencephalon is subdivided into metencephalon and the myelencephalon. So you have 8 different segments, and in this rhombencephalon, in the rhombencephalon, starting from R1 to R8, these are called as rhombomeres. Rhombomere 1, rhombomere 2, 3, 4, like 8. And each set of gene is distinct set of gene is required for each rhombomere development. So here these are represented here. And again, you don't have to remember all the genes for the rhombomere development. I'll be talking about what are the genes required for each rhombomere development in the next slide. So just to kind of overview, so you have these 3 segments, the rhombomere 1, 2, and 3. So these, these rhombomeres are developed from these sets of genes, which are shown here, like combinatorial exon of Hox A and Hox B, which is also color coded. So everything is color coded here, correspond to each rhombomere. So the trigeminal norm basically innervate rhombomere 1, rhombomere 2, and rhombomere 3. So these are again controlled by the homeobox gene. These nerves innervation is controlled by the homeobox gene expression. So the again rhombomere 4 and 5 is innervated by 2 distinct sets of nerve, which is the facial nerve and the abducens nerve. And in the rhombomere 6, 7, these are innervated by these sets of nerve. So if you have mutation, any sets of any kind of gene in this development, you have defect in the innervation of this nerve. So here, this is a wild type, rhombencephalon represented here in the B, and this is a mutant for the Hox A1 gene deleted, Hox A1 minus. So everything is normal. You have trigeminal nerve innervated, rhombomere 1, rhombomere 2, and rhombomere 3. You have facial and this nerve, abducens nerve innervated to rhombomere 4, rhombomere 5, and also glossopharyngeal nerve to rhombomere 6. So if you have deletion or mutation of this one of these Hox gene, the Hox A1 minus, in this you will have loss of rhombomere 4, and some of this, you have loss of the abducens nerve as well as the some of the nerve of the facial nerve are lost in these mutants. So that means the innervation of nerve and segmentation in the rhombencephalon depends on the expression of the Hox genes. Okay, so I've just briefly mentioned about everything here. In the next slide, this is weird, this is not moving. So in the next slide here, I am talking about another vertebrate. Previous slide, I talked about the development of the chick embryo, where I talked about rhombencephalon development by distinct sets of Hox gene. Here, this is an example of another model organism. This is the zebrafish. Okay, so you have the anterior person, you have the posterior persons. So the wild type, again, you have segmentation in rhombencephalon in zebrafish also. So you have starting from R1, R2, rhombomere, R3, R4, R5, R6, R7. These are regularly arranged in the wild type animal. But when you have in the mutation, when you have mutant of this gene, that is PBX gene. So when you have mutation in the PBX homeodomain gene, you have the loss of the rhombomeres. So you have loss of all the rhombomeres. Only the R1 is elongated and forms one rhombomeres. Got my point? So you have loss of some of the genes, some of the knobs that is innervated in the wild type animal is not present in this mutant animal. And these genes, the PBX gene, PBX2 gene, those genes, this gene is required for the rhombomere development in zebrafish. Okay. So any questions? So I just want to highlight that, the hindbrain, the rhombencephalon is segmented like Drosophila. You have the segmentation in the body. So the similar kind of phenomenon or similar kind of feature is also exist in vertebrate model system. So then just want to move to the next session of this of the lecture. So here in the previous lecture, I talked about in Drosophila, the expression of Hox gene depends on the expression of the upstream gene. Like when there is segmentation happens in Drosophila or cellular, you have the segmentation happens in Drosophila. First, you have expression of zygotic gene, the maternal genes. You have expression of bicoid and nanog or caudal. These are the maternal genes. Then you have expression of parule genes. First, the expression of gap genes, then you have that activates the expression of a gap gene, activates the parule genes. The parule gene activate the segment polarity gene and also the Hox genes. So those kind of mechanism exist in invertebrate like Drosophila, but in vertebrate animal, they use different mechanism to activate the Hox genes. So here, this is the retinoic acid that actually activates the expression of a Hox gene and the development, anterior-posterior development in the animals. So the retinoic acid is binds to the retinoic acid receptor, and it then it binds to the retinoic acid receptor in the cytoplasm. So enters into the cells and binds to the retinoic acid receptor in the cytoplasm. And that receptor, retinoic acid with the retinoic acid receptor, the entire complex translocate to the nucleus and bind to the retinoic acid responsive element to promote gene expression, to promote gene expression for the posterior development, to promote the Hox gene expression, so that you have you have expression of Hox gene and you have development of the posterior anterior posterior development in the animals. Okay, so here this is a normal embryo represented here. So you have the head and you have the tail, but when you increase the concentration of retinoic acid, it fails to develop the anterior part. It fails to develop the proper head. So that's how because retinoic acid is a posterior development signal. It defines the posterior person of the animals, or it specifies the posterior person of the animals. So so when you increase the concentration of retinoic acid, and you have loss of the anterior person, the anterior person fails to develop. So the thing is that I want to highlight, like in vertebrate, you have expression of distinct set of gene regulate the downstream gene, like expression of bicoid, nanog or caudal regulate the expression of gap gene, that in turn regulate the expression of parule gene, that in turn regulate the expression of segment polarity and Hox gene. So here the retinoic acid actually regulate the expression of Hox gene. That's how you have anterior posterior development happens in vertebrates. So here, as I mentioned before, that distinct set of genes required for the rhombomere development. Here, you don't have to again remember these genes. So here, these are the different set of genes I have listed. A lot of genes you don't have to remember. So like you have rhombomere 1 to rhombomere 2, like 7, 8. So here, these are the again color coded, which are expressed in which rhombomere is represented here. So some are highly expressed. Some are low, some are expressed not that much. So everything is represented here. Like for the rhombomere, let example, like suppose take example of rhombomere 2. So we have a lower expression of like this one, SIC1 in rhombomere 2. You have a higher expression of SIC1 in rhombomere 3. Again, you have expression of this gene in rhombomer 2, and everything is written here. So these gene sets of gene actually, these are the Hox gene, and they define the rhombomer development in animals. So you don't have to remember all the genes. Any questions till rhombomer development or rhombencephalon development? So that I can move to the next slide. Okay, so if there is no question, we'll move to the next section of the lecture. So then, what are the signals? We know that different set of gene is required for the anterior polarity or posterior polarity of the animal. Then, what these genes are producing, what are the signals? And there must be some kind of signal, some kind of morphogen that is defining the patterning or anterior posterior patterning. That is defining the head or that is defining the tails. So here is the set of experiment that is done in the Xenopus frog embryo to know what are the signals that defines anterior or posterior development. So here two sets of experiment are done. One is this is a Xenopus the frog embryo. So they have injected two distinct versions of the gene, that is, this is the truncated BMP receptor, and this is the dominant negative of the Wnt8 ligand, the gene, sorry. So we know that Wnt is required for the posterior development, and that we already talked about when we are talking about Drosophila. And these, when they inject the truncated version of the BMP receptor or dominant negative form of the Wnt. Dominant negative is the inactivated form of the Wnt, that we call as dominant negative form. So it suppresses the gene expression. So when they inject the truncated form of the BMP receptor or the dominant negative form of the Wnt receptor, you can see that, and these are the posterior, the Wnt actually defines the posterior polarity. So when they inject in the frog embryo, you can see that you have ectopic expression of, head, and you have ectopic expression of head because these, defines the posterior, the activated form of the Wnt or the BMP molecule, they define the posterior development. And here you are putting as an inactivated form, like dominant negative form or the mutated form of the BMP receptor. So that cannot define the posterior development. So that means you have more specification or more development in the anterior portion. That's why you have development of two head in the Xenopus embryo. Okay, any questions? So it's written there, I have everything here. So then they found out what are the inhibitors for the Wnt signaling pathway. So they did the genetic screening, and they found that, DKK1, so it's DKOF. So DKK1 is the, act as an antagonistic for the Wnt signaling pathway. So when they, when they also inject DKK1 mutant, or the the inactivated or the mutant form of the truncated form of the BMP receptor, you found similar kind of things, similar kind of two-head development, because DKK1 inhibits the posterior signaling, and BMP receptor, the mutated version of the BMP receptor, also inhibit the posterior signaling. So that's why you have development of two head in this version. Or this is the frog version of DKK1, and this is also again done with the mouse version of DKK1. And in this case also you see that you have development of two heads in the anterior portion, which suggests that, the signaling molecules, the posterior signaling molecule, the Wnt signaling or the BMP signaling, they are the posterior signaling molecule, and they define the polarity, which the head and tail portion of the animals. So this is the experiment people have done to, to say that Wnt is a posterior signal or BMP is a posterior signal, and this is the experiment they have done to define the posterior polarity of the Xenopus embryo. Another set of similar kind of experiment have done in the mouse model system, where this is the normal embryonic development of the mouse. This is the normal mouse, and this is the mutant version of this. So you have These are the different bones present in the head portion of the mouse. You have maxillar bone, you have mandibular bones, and you have also the parietal bones. So this is the normal mouse where you have normal expression of DKK1 that I talked about in the previous slide, or the noggin. We know that noggin as an inducing molecule, which induces to form the neurons. So these are the wild type mouse. So you have formation of normal formation of these bones. But when you have mutation, either of these two, like DKK1 mutation or noggin mutation, so alone everything seems fine. But when you combine these two, like DKK1, one copy of DKK1, or another copy of noggin mutant, you find that you can see that those bones are missing here in the mutant. So you don't have the formation of maxillar bone or mandibular bones in this animal. That suggests that there is a synergistic interaction in the BMP and Wnt antagonist for the head development. So noggin is antagonist for the BMP signaling pathway, and DKK1 is identified as antagonist for the Wnt signaling pathway. Okay, so there is a synergistic interaction between the BMP and Wnt antagonist for the development of anterior polarity, or for the development of a head in the mouse embryo. Any questions? Yes. Yeah, so here just I want to highlight that like the synergistic interaction between the two signaling component and how it defines the posterior and anterior development in the animals. So in the upper panel, you have the normal expression of DKK1, which is antagonized for the Wnt signaling pathway, and noggin is antagonized for the BMP signaling pathway. So here you have normal bone development because everything is fine here. So you have parietal bone, you have maxillar bone and mandibular bones, which are normally developed. So when you have you have loss of one copy, like in the two chromosomes, you have loss of one copy of DKK1 and also another copy noggin 1, then the bones are not developed properly. So that suggests there is a synergistic interaction in the antagonist, like the BMP antagonist and the Wnt antagonist for the development of animal, the posterior development of the animals, posterior and anterior development of the animals, because we know that Wnt regulate posterior development, right? So, and this antagonist DKK1, it inhibits actually posterior development. Is it clear or? Okay. So besides this signal, the expression of homeodomain genes also defines the posterior and anterior development in the animals. So this is shown here. So you have the forebrain, you have the midbrain and the hindbrain, and you have the spinal cord. So for the development of anterior person, you have expression of OTX2. So OTX2 actually expressed in the actually defines or specifies the anterior portion of the during development of the brain, and it expressed throughout the brain development. Throughout the development, it expressed. However, the GBX2 it defines the posterior development mostly. It defines the hindbrain and as well as part of the spinal cord. And OTX2 defines the midbrain and specifies the midbrain and the forebrain development. So these are the sets of genes that you want to remember, that which gene is required for the midbrain development or forebrain development, and which genes is required for the hindbrain development or the spinal cord development. So just have the two sets of gene. OTX2 is required for the forebrain and midbrain development, or GBX2 is required for the hindbrain and spinal cord development. Okay, so now here, when you have in mice, so in Drosophila, the OTX2 is homolog of Drosophila gene orthodendricle, is the same gene is present in Drosophila, which is required for the hair, development of the head structure. So in mice, knockout for the OTX2 leads to the loss of the forebrain, like the prosencephalon, the forebrain. So in the mice, you can see that the forebrain is not developed when the OTX2 is lost. Similarly, the GBX2 knockout mice, which specifies the hindbrain, the rhombencephalon. You have a loss of a GBX2. You find the loss of this rhombomer like from R1 to R3. So this is just to remember that OTX2 defines the anterior development of the brain like midbrain and forebrain, and GBX2 defines the posterior part of the brain like the rhombencephalon, the metencephalon and myelencephalon, the posterior part of the brain, the hindbrain. Okay, so this is how the expression of a homeobox gene, the expression of different signal like Wnt signal defines the anterior posterior polarity during animal development. So this is second kind of so the head development is not only one process, it is a complex process. There are a set of genes required for the head development, the brain development. There is a set of signal required for the brain development, and there are certain region which is also crucial for the head development or the brain development, mostly the brain development. And here again the two distinct stages of the brain. This is stage 10 and this is adult brain. So you have the different segment of the brain, the prosencephalon, mesencephalon, and you have metencephalon and rhombencephalon. So like as I mentioned, different signal, different genes required for the development of the brain. You have distinct center called as organizing center in the brain, which is required for the brain development. And this organizing center is called as it is present in the in between the border between the mesencephalon and metencephalon. So this is midbrain, hindbrain border. So what people have done is, so here everything is in kind of this part. Prosencephalon develops into forebrain, then mesencephalon develops into midbrain, which and here you have metencephalon develops into cerebellum, and rhombencephalon develops into medulla. So in the adult stage, so this junction is crucial for also for the organization of the brain or development of the brain. So what experiment people have done is, when they have in the stage 2 embryo, stage 10 embryo, when they have taken part of this metencephalon here, and they put in close to this organizing center, and when they take this part and put it in the forebrain, and they found that there's ectopic expression of midbrain because this tissue is derived from the midbrain and the hindbrain border. Okay. So there is ectopic expression of midbrain and also in the cerebellum, which is shown here in this cartoon, and that means that something present in this in the organizing center, which is inducing the formation of this structure, and which is also represented here. So here you can see that you have expressed, this is the midbrain and this is the ectopic expression of ectopic formation of midbrain. That happens because this tissue or this is excised from here and put it in the telencephalon or in the forebrain. That actually those signals, something is there in that region, and those signal is inducing the formation of ectopic formation of midbrain or the cerebellum. Okay, so you require distinct set of a gene, you require distinct set of signal like Wnt signal or BMP signal, and also you require the region, the organizer, like as I mentioned, in the Drosophila development, you have the organizer present in the Drosophila gastrula, in the gastrula stage. You have animal like in the amphibian, you have same kind of organizer present in the amphibian development. Similarly, in the vertebrate, like in the higher organism, you have the organizing center in the brain, which is required for the brain development. So it is a complex process. Require signal, require set of gene expression, require a specific region for the development of the brain. And what are the signals? I will be coming soon. So any questions in this part? Okay, so this signal is written here. This is FGF8 signal. this signal is critical for the organizing activity, and that is present in the meson and metencephalon boundary tissue. Okay, so I'll show some more experiment people have done for identifying the organizing center. So several signaling molecules such as Wnt1 or FGF8, the FGF, the fibroblast growth factor, FGF8, are localized in this junction, the midbrain hindbrain boundary. Okay, so this is a normal mouse embryo. So you have telencephalon, which is part of the forebrain. You have midbrain, you have a hindbrain, you have a spinal cord, and the Wnt, it shows that the expression of Wnt is required for the development of the brain. And this is another signal that I mentioned, that the Wnt1 and FGF8 is present in the organizing center, that is organizing or that is regulating the brain development. So here this is regulation of brain development by FGF8 signal. So here when you have loss of this Wnt1 or Wnt1 in the organizing center, you can see that this is the normal mouse embryo, embryo in the mouse. So you have diencephalon, you have telencephalon, you have melencephalon, you have cerebellum, you have midbrain. Everything is there in the normal mouse embryo. But when you lost, when a mutation of Wnt, or when you don't have, you restrict the expression of Wnt, then you can see that you have loss of the midbrain and cerebellum in this in this mouse embryo, the mutant mouse embryo, which suggests that the organizing center, in the organizing center, those signaling molecule, the FGF8 signaling molecule or Wnt1 is necessary, is required for the development of the midbrain or the development of the brain. Okay, so everything is written there. Like I have just to go, so it will be easy for you to go over the slide and kind of you don't have to search anything. So everything is written there. So now, if I summarize about this organizing center in the model, so this is a simple model that is represented here. So these are different stages of the embryo development in the mouse. So this is the early development of stage like E7.5 or and this is the mid 8.0, 8.5, and this is the latter 9.5 embryo. So what happens is the two transcripts, the homeobox transcription factor, that is OTX2 or GBX2, they define the anterior and posterior polarity of the brain. So OTX2 is required for the anterior development, like the midbrain and forebrain development, and GBX2 required for the hindbrain development, the rhombencephalon development. So first you have the in the early stage of the embryo, you have polarity, anterior posterior polarity. So you have expression of either you have expression of OTX2 and you have expression of GBX2. Then after that, there is a development of this organizing center at E8.0 to 8.5 embryo. So you have development of this organizing center, and from this organizing center you have the secretion of this signal FGF8 or Wnt signal, which is represented here, that defines the again anterior polarity or posterior polarity of the embryo. So everything is written there, and these are the signal which actually kind of, that is how you have development of the brain. So you require three things is happening here. You require expression of genes like homeodomain genes. You require is a combinatorial effect for the brain development. You require the signals, and also you require the organizing center for the development of the brain. Okay. So if you are clear about this hindbrain development or the rhombencephalon development, then we'll go to the forebrain development, which is the prosencephalon development. So there is also segmentation. Different set of gene is also expressed in a different compartment for the forebrain development. Any questions for the hindbrain development? Okay. So if you have no question for the hindbrain development, we'll move forward to the forebrain, the prosencephalon development. So here there are two different stages of the development represented in this slide. So it is the mouse embryo 8.5 stage, and this is 10.5 mouse embryo. So in 8.5, so this is the forebrain, this is the midbrain, and this is the hindbrain. So we talked about the rhombomer development from then we are going to talk about this prosencephalon development. So again, these are segmented, so which is represented here clearly in the latter stage of the development. So prosencephalon is divided into two distinct compartments. You have a telencephalon, this compartment, and you have diencephalon. Here is the mesencephalon, here is the rhombencephalon. So in the telencephalon, this part, you can see that you have two distinct hemisphere. The anterior half hemisphere, which is defined by EMX1 gene, and in the posterior half hemisphere, which is defined by EMX2 gene. And each segments is called as prosomeres, and P stands for here prosomeres, and R stands for rhombomeres. So here, so you have in the telencephalon you have prosomere 3, 4, 5, 6. And in the diencephalon you have prosomere 1, prosomere 2. So again, those genes actually define the specific boundary of this in the prosomere development. Okay, so and these are homeobox genes or homeodomain genes. So here I have just mentioned that the prosomere 9 is required for the eye development, which is similar to like eyeless in here in Drosophila. Okay, so here just example showing that eye development. So when you overexpress like PAX6 or eyeless, so the prosomere, as I mentioned, there are 9 prosomeres in vertebrates, prosomere 1 to prosomere 9. So prosomere 6 required for the eye development, which is called as eyeless in Drosophila. So when you overexpress the PAS6 or eyeless in Drosophila, you can see that there is an ectopic. This is the normal eye, and there is an ectopic formation of eye from the leg. Similarly, in Xenopus embryo, when you express like PAX6, you have ectopic expression of eyes, which is in the arrow. So that means this is how this anterior polarity or forebrain development happens by the expression of distinct homeobox genes, PAX genes, in the in the anterior portion of the animals. So any question in the forebrain development like PAX genes? Okay, so this is kind of just to give you an overview that how the brain develops and what set of gene and signal is required for the brain development. So again, in the forebrain you have two distinct compartments. You have EMX1 and EMX2 in the telencephalon. Like in the previous slide, you can see that you have in the telencephalon you have two distinct compartments, two distinct hemisphere, anterior half hemisphere, which is EMX1 and EMX2, the posterior hemisphere. So EMX2, so EMX2 and PAX6. PAX6 actually expressed in the posterior part. PAX6 is expressed in the anterior part of the brain, and EMX2 defines the posterior part of the brain. Okay, in the posterior cortex, and this defines the anterior part of this cortex. Okay, so this is a normal wild type mouse. And you have regular arrangement. You have motor cortex, which is regularly arranged. You have a somatosensory cortex. You have visual cortex representing the blue, light blue, and you have also auditory cortex. So this is how the normal wild type mouse looks like. And these are the genes EMX1 or EMX2. Here I am talking about EMX2. So it defines the visual cortex EMX2, and the PAX6 defines the motor cortex. So in the mutant EMX2, you can see that you have you have reduction in the visual cortex. You have more specification of the motor cortex, and the opposite thing happens in PAX6. So in the PAX6, you have less development of the motor cortex. You have more development of the visual cortex. So this is how these genes are required. The PAX6 genes or PAX genes or prosomere genes or the EMX1 or EMX2 genes, this is how the genes are required for the development of the forebrain, like EMX2. By the combinatorial action of EMS2 or PAX6 or prosomere genes, you have patterning in the forebrain. Okay, so now if I so I talked about what are the genes required for the forebrain development. Then we are going to talk about what are the signal required for the brain development. Okay, so you require two distinct set of signal for the anterior development and posterior development. So in the posterior development of the brain, you require in the forebrain, you require the BMP and WNT signals for the posterior development of the forebrain, and you require the FGF in the for the anterior development of the forebrain. Okay, so this is a control condition where you have normal localization of all of these region, like you have anterior specification, you have posterior specification, you have BMP here in the posterior part, you have FGF8 in the anterior part, you have somatosensory region present in the middle of this embryo, or in the middle of this developing embryo. But when you add upregulate one kind of signal, suppose you put FGF containing beats in the anterior part, you have more specification of the anterior head like cortex region. Then the somatosensory cortex, which is present in the middle portion of this, it moves to the posterior region. Okay, and when you put the FGF8 containing bead to the posterior portion of the posterior portion of this embryo and the brain, and you can see that the somatosensory cortex now divides into two. There is ectopic expression of somatosensory cortex. So, so the thing that I want to highlight, that you require for the for the cortex development, you require a distinct set of genes and also distinct set of signals, like FGF signal, you require the WNT signal and the BMP signal for the development of cerebral cortex in vertebrates. Okay, so so this is all about the anterior posterior patterning in vertebrates. So any questions from this anterior posterior patterning? Then we'll go for the dorsal patterning. So the take-home message for the anterior posterior patterning is that you require distinct set of signal, you require distinct set of gene, and you require the organizing center, which express again distinct set of signal, like FGF8 signal or WNT1 signal for the development of for the patterning of anterior posterior patterning of the brain. Okay, now we'll move to the next section, which is dorso-ventral patterning of the vertebral neural tube. So so this is just want to show that how neural tube looks like after closure. So here after closure of during after gastrulation, you have neural tube closure. So these are different structure present in the neural tube. So you have, this is the neural tube, and you have the epidermis, and between the junction of neural tube and epidermis, you have distinct set of multipotent stem cells which arises from this in this region in the junction between neural tube and epidermis. These are called as neural crest cells. Okay, and this is the neural tube and this is the notochord. So what happens is when it develops, the neural tube develops further development. During the further development of the neural tube, you have initiation of certain structure. Okay, first you have initiation of the foreplate in the ventral region of the neural tube, and you have initiation of roof plate in the dorsal region of the neural tube, and also between this axis, anterior posterior axis, the dorso-ventral axis, you have formation of another kind of group kind of structure called as sulcus limitans, and these structures are important for because they contain distinct set of signals for the development of dorso-ventral patterning of the neural tube, so which we'll be going to talk about soon. So the neural tube again differentiate into matured structure, and here the notochord, it is a mesodermally derived tissue. Okay, so this is during the gastrulation phase. You have from the neuroectodome neuroectoderm invaginates inside, then it forms the mesoderm. So this is a mesodermally derived structure, the notochord, and this actually forms this foreplate, and also it forms these nerves. These are the spinal motor neurons. So those are derived from this. Those are notochord gives signal so that they form these cranial nerves, the spinal nerves, and also it forms the floor plate. So this is a set of experiment people have done to show the importance of notochord. So in this condition, when notochord is lost or notochord is not there, you don't have any formation of floor plate, and also you don't have formation of spinal motor neuron. But when you put, or when you transplant the notochord in this axis, like dorso-ventral axis, another side, when you transplant this notochord, you can see that you have formation of floor plate, and also you have ectopic formation of motor neuron, which suggests that the notochord plays an important role for dorso-ventral patterning or development of the spinal motor neurons. And we'll come to know what are the signal that notochord contain, which is actually specifying this structure, or this like the floor plate or the ectopic expression of in the motor neurons. Right? So so those things are mentioned here. So here this is a simple experiment people have done. So you have in the dorsoventral patterning, in the ventral side you have the foreplate, which is formed by the notochord, and you have the roof plate, and here you have a sulcus limitans. So this is the dorsal side, this is the ventral side, and this is the intermediate portion of the developing embryo. So here what kind of signal they want to identify the notochord contains. So the notochord contains the ventral signal, which is sonic hedgehog. So they here here they have done the experiment when they added the sonic hedgehog. When first what they have done is they have taken some part of this explant from this region, intermediate region, and they cultured two distinct set of culture they have done. Here in this culture, they did not add sonic hedgehog, and you can see that you have expression of PAX7 genes, which which defines the dorsal segment, which defines the dorsal patterning of the animal. And when they added the sonic hedgehog, you can see that you have expression of these ventral genes, which are NKX2.2 or oligo2. So here is the experiment. So when they add the sonic hedgehog and the increasing concentration of sonic hedgehog, you can see that the posterior segment posterior genes, which defines not posterior, the dorsal genes, the PAX7 gene, there is a reduction in the expression of PAX7 gene because you are adding the sonic hedgehog to the culture, which actually defines the ventral differentiation of the neural tube. That's why you have a reduction of the dorsal and the genes which defines the dorsal differentiation. Now you can see that with increasing concentration of sonic hedgehog, you have more expression of oligo2 or NKX2.2. So so you can say that the sonic hedgehog is a ventral, it differentiates the ventral patterning of the animals. Okay. So the sonic hedgehog shows a polarizing activity, and these sonic hedgehog basically secreted from the notochord to differentiate to initiate the ventral differentiation of the neural tube. Okay. So now this is a slide to talk about different set of tissue or different set of cells which are also generated during the dorsoventral patterning. So if I come to the previous slide, here you have the neural crest cells, which is a multipotent stem cells present between the epidermis and the neural tube, and those neural crest cells actually develops into other types of tissue, other cell types during development of the brain. So this is again the diagram showing the chick embryo. So you have prosencephalon, the forebrain, the mesencephalon, the midbrain, you have rhombencephalon, the hindbrain. Now you have the spinal cord. So you have like from S1 to S28. These are different segments of the spinal cord. So those neural crest cell basically, forms the different set of neurons like mesoderm or the pigment cells or the sensory ganglia. So those are actually, generated by the, from these multipotent stem cells, which is the neural crest cells. Again, you don't have to remember all these things which are mentioned in this slide. Okay, so this is how you have the dorso-ventral patterning happens during development of the embryo. So you have secretion of sonic hedgehog from the notochord, and also that defines the ventral, that express the ventral set of the genes that is required for the ventral development. And also you have you have the generation of different set of cells or different set of these things that are mentioned here, cells from the neural crest. So that's how you have development of dorsal nerve tube. That's how it is developed during development of the animals. Okay, so another set of the experiment with sonic hedgehog and also with BMP people have done to identify what are the dorsal genes, what are the ventral genes required for the dorso-ventral patterning. So again, the same thing is shown here. You have neural crest cells, which defines which forms different sets of cells like ganglion cells. You have other cells which are mentioned in the previous slide, and here you have the formation of the floor plate from the due to the activity of notochord. You have formation of a roof plate, and what they have done is you have dorsal side, you have ventral side. What they have done is again they have done experiment, similar kind of experiment here to show that how BMP, the bone morphogenetic protein, regulate the dorso-ventral patterning in the animals. Okay, so here the explant is taken from the intermediate part of this, the junction between ventral and dorsal part of this called as intermediate part. The explant is taken from this part, and they are cultured here. The tissue is cultured here without the addition of ectoderm or BMP signaling molecules. Okay, so so here they have taken two distinct morphogen. One is the sonic hedgehog, and another is the BMP, increasing concentration of BMP. So when they put increasing, when they put the increasing concentration of BMP, so either they took the ectoderm that contains the BMP molecule, the ectoderm has the BMP signaling molecules. You have expression of these genes, and or they added and they later found that the BMP4 and the BMP7 basically required for the expression of these genes, which is is a posterior signal. So this is the BMP is a posterior signaling molecules. So when they added the increasing concentration of BMP, you can see that you have expression of the MX1 or NKK 2.2. You have down regulation because this is a ventral patterning genes. So when you have increasing concentration of BMP, you can see that is down regulated, and this gene is up regulated because this is the posterior, not posterior, the dorsal the genes that is required for the dorsal development, the PAX, either PAX6 or MX1 or 2. So this is how the sonic hedgehog and BMP that defines the signal that defines the polarity of the neural tubes. Okay, so we'll come to the last slide. Last slide, just to summarize how the signal interaction of different signaling molecules define the dorsal or ventral polarity during development of the nervous systems. So first you have after gastrulation you have notochord formation. Then the sonic hedgehog, first you have activation of sonic hedgehog. The sonic hedgehog secreted from the notochord that defines the ventral polarity, and it transcribe the genes required for the ventral development of the nervous tissue. Now after that you have the dorsal initiation of other signal, which actually releases from this roof plate, which is the BMP signal, which defines the Wnt signal, which is dorsal signals. So this is how, and that forms a gradient. So you have more signal here in the BMP or the Wnt, and it acts as a morphogen. So it acts as a gradient. So you have more expression of the BMP or Wnt in the dorsal side, and that goes down when it comes to the ventral side. So this is how the interaction of either dorsal signal and ventral signal. This is how the dorso-ventral patterning happens during the development of the neural tube. Okay, so the two signal, this is how this happens, during development. So you have initiation of the first, you have expression of sonic hedgehog, the ventral signal from the notochord. Then you have development of the ventral part. Now you have expression of the BMP or Wnt from the dorsal region, and you have specification of the dorsal part. So this is how the two signal antagonize one another. The mutual antagonism is required for the dorso-ventral patterning of the nervous system. Any questions from this dorso-ventral patterning? Okay, so this is the end of this lecture. So we finish the patterning of the nervous system, and next section will talk about neurogenesis. Thank you.